Welcome back to The Center. This is a podcast from the Center for Neurotechnology and Neurorecovery at Massachusetts General Hospital. I'm your host, Josephine Bucles, and today I'm joined by Dr. Anastasia Yendiki and Dr. Chiara Maffei. Dr. Anastasia Yendiki received her PhD in electrical engineering from the University of Michigan. She's an associate professor at the Martino Center at Mass General Brigham Hospital. Dr. Chiara Maffei received her PhD and master's degree from Trento University in Italy. She received her PhD in neuroscience. She is also an instructor at the Martino Center at Mass General Brigham. So, Anastasia, can you tell us what tractography actually is?
Yeah, so tractography is a computer algorithm that takes the diffusion MRI scan, and the diffusion MRI scan gives us information at every location in the brain. How are the fibers oriented? Which way are they pointing? Mm-hmm. And so a tractography algorithm follows those vectors from point to point in the brain, sort of connects the dots, following that path, so that starting from one point, you get to all the other points in the brain that are connected to that.
And how did you get into this very complicated field?
Yeah, so it was serendipity, really. Okay. So I first came to the Martino Center as a postdoc to learn about functional MRI, because that's one of the things that the center is known for, as one of the first places that did fMRI studies. And what happened was, actually, when I interviewed, one of the people I spoke with showed me a picture of tracts from a diffusion MRI scan on his screen, and explained how the way you get this beautiful picture of all these colorful bundles is that there are water molecules that sort of travel across these along these bundles, and that with MRI we can image those microscopic movements and go from those microscopic movements by connecting the dots to the whole brain connections, and that just blew my mind. So I thought that was the most science fiction thing I had ever heard in my life.
I mean, that's like the rainbow map that we all are familiar with. The brain.
So then I started, coming up with a game plan for how I was going to shift.
Transition.
Yes.
That's wonderful. That's a really exciting story, because I think that's like what interests us all in tractography, is that like kind of rainbow image of the brain being connected.
Yeah, that draws you in, and then you get into all the complexities, the nuances that we were talking about.
Chiara, can you tell us about your origin story?
Yeah, I think I have a similar story.
Yeah.
I was doing my master's in Trento, and I think it was an MRI physics class. Mm-hmm. And it was the first time that, they were talking about functional MRI and diffusion MRI, and someone showed this colorful tract. And I really liked the idea of mainly doing something that not everybody else was doing, because back in the days, everybody was doing functional MRI. Yeah. Like, there was a very long line. And I often try to knock you where there's already a million people, because it doesn't seem very effective. Yeah. So it's like, oh, this seems interesting, and nobody else here is doing it. Let me see if I can, step into this. And that's actually how it happened.
So one really exciting study that you both were on was looking at a kind of decomposing one of a big cortical pathways in the brain, the SLF. Can you talk about some of the techniques you were using to kind of validate our current metrics for tractography, and how we were looking at it from a more granular perspective?
Mm-hmm. So that was actually work led by Chiara, so maybe I should let her describe it.
Okay.
So it's interesting that we started our work thinking that we would get somewhere, and then we got somewhere completely different. So there are certain long-range connections in the brain that travel long distances from one end of the brain to the other, and so there are many opportunities along that path for tractography to get things wrong. Let's say to stop prematurely. And so we thought that if we went to higher and higher resolution, we would be able to see these long-range connections uninterrupted, to see more of these uninterrupted long-range connections that go all the way from one end of the brain to the other. But what happened is when we went to those high resolutions, we actually saw that these long-range connections actually became decomposed into shorter range connections that you can think of as side roads that merge into a big highway and merge on and off and connect different pairs of more nearby regions rather than going all the way from one end of the highway all the way to the other.
That's really exciting.
And that replicates things that anatomists actually knew for many, many years. But in the diffusion tractography world, we had been used to thinking of these pathways as one monolithic structure that goes all the way from one end to the other. Because as you go to the lower resolutions that are typical of an in vivo diffusion MRI scan, where you can put a patient in the scanner for, let's say, 10 minutes and get an image, then at those lower resolutions, all these smaller pathways sort of merge into one big pathway.
And so Anastasia mentioned in vivo imaging. That was one of the methods that you used. What were some of the other methods that you used?
We combined in vivo at different resolutions with ex vivo imaging, much higher resolution. Also with tracer data, macaque brains, and optical imaging. So we really had like both human data across species, modalities, and resolutions.
And so was the goal for this research to kind of map the SLF, or was it to validate in vivo diffusion imaging?
As Anastasia we started off thinking of how we could validate the SLF using higher resolution and showing what the real anatomy was. We ended up doing the same, but in a different direction. Okay. So we showed that the anatomy was actually different from what we thought it was going to be, and that the higher resolution we go, the more granular things we can see. And what was very nice was to see that this was replicated across species and modalities. So this was true for the optical imaging, it was true for the tracer data, and it was true across the species.
And species, you're referring to humans and macaques.
Macaque,
Okay. So you mentioned ex vivo imaging. That's not something that I'm familiar with. Can you talk a little bit about that?
Yeah, so in ex vivo imaging, we can use samples, post-mortem samples, and we can put them in the scanner. And the great things about this is that we can leave them in the scanner for much longer than we would be able to leave a live person.
They don't complain.
Okay. They don't move either, which is another great plus. But yeah, so this allows us to get to much higher resolution, retaining a good SNR, great SNR of the images. So
what is SNR?
Signal to noise ratio. That's what it stands for.
Okay.
But what it actually means that we can still see things very well. We can have clear images of what we're seeing.
Okay. So in an ideal world, a person would be able to stay in a scanner for how long to get the best SNR?
Forever.
Okay. Okay.
Well, we're getting better. Scanners are getting much better.
How long were you keeping the brains ex vivo?
So we might leave them, in a typical scenario, we'll leave them over the weekend.
Okay.
Yeah. Okay. Because to do these kinds of scans, we have to use the time that nobody else wants to use.
Yeah, that's fair.
So we would typically put in the brain in the scanner Friday afternoon and then
come back
it up on Monday morning. Although more recently we have done some more extreme acquisitions where we wanted to get whole human hemispheres imaged at very high resolution, and we scanned them for a whole week.
Wow.
Yeah.
Wow, that's amazing. I bet that costs a lot.
No comment.
You mentioned also optical imaging and tracer. Can you talk a little bit about those too?
Yes. So they give us different information. So the optical imaging, we can do it in humans. So it's in postmortem human tissue. The tracer we can only do it in animals because it's an active tracer. It means that it actually binds to the axon when the animal is alive.
Yeah.
And so that we can actually see all the entire trajectory from an injection point, to
all the way back.
This axons are going in the brain. So it gives us information about the entire trajectory of a path. Whereas with optical imaging, we can get the local information of these fibers, and we can have that information in humans, which is great to be able to combine these two types of information.
And there's also another method called X-ray imaging as well. Can you talk a little bit about that? Was that part of this study?
Not this particular study, but in a new project that we have that I think we're going to talk a little bit about as part of the Brain Connects program, we are collaborating with a team at UCL that scans whole human brains in a synchrotron. And these experiments currently can only be done in one place, in this one synchrotron, the European synchrotron, which is in Grenoble in France. And they have a special X-ray beam, an ultra-brilliant beam is what they call it, that allows them to image whole human organs down to resolution of a few microns. So that is an even more experimental technique than some of the other ones that we talked about later that have been in use for a little bit longer.
And is that somewhat similar to like a method like electron microscopy?
So we can't quite get to the resolution that electron microscopy would get to, because that's at the nanometer scale. Right now, those kinds of acquisitions with electron microscopy at the nanoscale, so far they've been done, a large volume for that kind of imaging would be 1 millimeter cubed. And there are other teams within the greater Connects program, not us, that are trying to scale up those methods, but to image, let's say, parts of the mouse brain. So those kinds of methods cannot at this time scale to the big brains, the human and non-human primate brains.
Okay. I guess like coming back to this study, because I think it's really special that there were so many different techniques that were being used. And essentially you showed that like the diffusion tractography wasn't as granular as it could be. What are some of the ways that you can see this like impacting the way that we use tractography in clinical settings?
Yeah, I think this specific study is actually great from this point of view, because I hope it will help change the way that we think about these pathways. So as Anastasia said at the beginning, because of the way tractography works, we have been used to see these uniform, shiny pathways. But that's probably not really reflective of the true way of how these things are organized in our brain. And so hopefully it will like force us to rethink about how these pathways are actually made of many subcomponents, and how each of these subcomponents might be involved in different functional networks rather than being one single thing involved in one network, or how these different subcomponents might be impacted differently in different diseases, for example.
Yeah, it's interesting because we had a conversation recently about the difference between structural connectivity and functional connectivity, and how tractography sometimes is able to, show structural connectivity, but isn't able to actually mimic functional connectivity. Do you think that separation is going to be brought closer together with more granular approaches?
Yes, I think so.
Okay. It's exciting.
That's it. With tractography, we get a map of the roads, and functional imaging gives us the traffic, which will change on the same road depending on what time of day it is.
It's great. That was the exact analogy that was used to describe the difference, to help explain it to me. So I think it's wonderful. So you mentioned the Brain Connects project, and we kind of like as lay people are aware of the connectomic space through the Human Connectome Project as this like large scale effort to combine a bunch of data, combine a bunch of MRI data, to develop a good like gross tractography map for humans. Can you talk a little bit about how the Brain Connect group differs, and like how your goals are different from the Human Connectome Project?
Yeah, that's a great question. So both are large-scale data collection efforts, and both are trying to probe connections in the brain. The goal of the Human Connectome project was to scan as many subjects as possible with very high quality in vivo MRI scans. So there the goal was how to scan a very large population to be able to see individual variability, for example, and to be able to do studies that require a large number of subjects. So, for example, if you wanted to look at association between different behavioral measures and brain structure or function, the Human Connectome project gives you a large data set where you can try to tease apart these small effects. With the Brain Connects, the goal is not to scan a large number of subjects, but focus on a small number of postmortem specimens and try to get the highest resolution possible images from those samples. So try to get the most detailed map of brain connections to date from these small number of specimens, and try to figure out how we can scale up those techniques that are required to get these super detailed maps, so that maybe in the next phase of the program, if we're lucky and it gets continued, we can actually apply them to a larger number of brains. It's not going to be, the thousands of brains that were scanned for the Human Connectome project, but if we could do this in, let's say, 100 brains, it would give us some better idea of, inter-individual variability, or we could look at disease versus control brains.
Okay. And so currently right now it's healthy controls that you're using. And how many patients is that right now?
So it's going to be somewhere like around a 10, brains from each species in our project.
Wow. And what techniques are you using?
So it's...
Many.
Yes, the kitchen sink. So because there isn't a single imaging modality that can give us all the information that we want, we are using a number of techniques that are the most promising. At least that's how we felt when we wrote the proposal, and we still feel. And so we first do diffusion MRI scans on the intact brains. That's the first thing that we do, because that gives us the link to the in vivo neuroimaging, the technique that we can actually hope to do in actual living patients. And then all the other techniques are techniques where we eventually have to slice and dice the brain and then put the data together. So we're using both optical and X-ray microscopy, some of the methods that were mentioned before, and it's the first time that we're trying to do this in such large samples. So some of those techniques have been done, let's say light sheet microscopy in an individual section from a brain, or some of the other methods in sort of smaller volumes inside the brain. So we're trying to scale these up to a large volume of the brain, large enough to give us some meaningful connections that are clinically relevant, and develop accurate models of the pathways that are relevant for neuromodulation for motor and psychiatric disorders.
Neuromodulation is in DBS.
Yes, exactly. Deep brain stimulation.
Okay. And how could this research apply to DBS?
Do you want to take that?
Yeah,
that's
sure.
Another work that she's leading.
I can, yeah. Yeah, so DBS works. It's been proven more and more that DBS is very effective when the white matter pathways are actually stimulated rather than the nuclei themselves.
Wow, okay.
So that's also why there's been a lot of interest recently in combining tractography with DBS. But what it's important for us is that the region that is most often targeted for DBS, specifically for Parkinson disease, for example, is located in a very complicated, dense area in the middle of the brain, where there's many small pathways that sort of like converge and are very close to each other and very close to these other nuclei. So conventional tractography cannot really, tease all these different components apart. And that's why neurosurgeons a lot of times rely on maps that come not from tractography, but maybe from other sources, like, old neuroanatomical atlases. But thanks to the high resolution tractography that Anastasia mentioned before, where we scanned for a whole week ex vivo, we could reconstruct all of these small pathways. And we're now about to release this atlas. And we hope it will be useful.
That's very exciting. One thing that is really interesting that you mentioned was like targeting white matter track pathways versus targeting nuclei. Are you saying that there's more success actually targeting the pathways rather than the nuclei themselves? Do we understand why that's the case?
I don't know much about DBS, but what I know is that the frequency that they are using to stimulate, it actually is more effective on the white matter pathways. And this is how far I can go with my understanding.
No, that's really interesting. I think like we're taught that it is like the main targets are in specific regions, but like nuclei specifically, and not like gray matter, but not white matter. So
that's why it's led, I think, to a sort of a rebirth of interest in connectional anatomy.
Definitely. And so DBS is like it's been very well validated in Parkinson's disease. I think it's almost ubiquitous. And you mentioned psychiatric disorders, and I mean, I guess other movement disorders. Can you actually talk a little bit about some of the psychiatric disorders? Because we have in the past talked a little bit about applications of DBS for depression, but I'd love to hear about how your research is kind of coloring that work.
Yeah, so it's interesting that in the disorders where this has been most successful and where there's more consensus about which particular connections to stimulate, like motor disorders, the connections are actually much better known and much simpler. So it's mostly connections that come as a straight shot from, the motor cortex all the way down to the brainstem, whereas the connections implicated in psychiatric disorders are the ones that project to and from the frontal cortex. And so they have to travel through a much more complicated areas and have to travel all the way from, the front of the brain all the way down. And those connections are harder to tease apart with conventional imaging techniques, with in vivo neuroimaging. And so there's also more controversy about what exactly these connections look like. And we're actually very lucky to be collaborating with a neuroanatomist, Suzanne Haber, who is the world's expert in some of these connections and has been doing these kinds of tracer studies that Chiara mentioned for decades. And so we actually, that's the this work has been really inspired by the studies that she has been doing.
It's exciting. I want to ask about how you model psychiatric diseases in macaques, specifically with tracers.
So it's we basically just want to map the connections. So again, it's similar to the idea of mapping the connections first in a healthy subject. So we don't assume that the macaques actually have any of these disorders.
There's no behavioral.
Okay.
Right, yeah. So the idea is to just get a clear picture, get the ground truth of what part of the brain connects to what.
Okay.
Yeah, and so I think if I can add to what she's saying before, between motor and psychiatric, I think some of the difficulty there is also related to how much we know about the function,
right?
The functional, involvement of this too, because obviously we know much more about motor systems and how those work, but it's much harder to know how actually OCD, works in the brain, what is going on. It's much more distributed. So I think that complicates, the task of deciding which pathways need to be stimulated versus motor.
So currently, right now, you're working only with healthy controls to kind of create structures and tracks of the brain. Do you foresee these being different in patients with movement disorders, patients with psychiatric disorders, and by how much will these differ? Will these maps differ?
So We expect that certain connections exist in every brain.
Mm-hmm.
So it's not like patients with these disorders will be, missing, a main connection that exists in the control brain. But we do expect that there will be differences in the strength or the relative strength of these connections between the patients and the controls.
Okay, that's interesting. And can that strength be reflected in the tracing studies and in the optical imaging and the X-ray imaging?
That actually, what's really good for reflecting it is actually diffusion MRI.
Oh, okay.
So the studies that we do will allow us to make a very precise map of the network of roads, so that then we know what specific ones to focus on, or when we see in the reverse order, actually, when we see a difference in the patient's networks, we know exactly what pathways go through there. So there are quite promising biomarkers that we can get from diffusion MRI, in addition to getting, the connections in the brain that reflect changes in the microenvironment and the microstructure.
And does fMRI play a role in this?
We don't really
We're
get
not
involved.
interested.
Not
Okay.
interested. We don't do fMRI.
Okay, that's really interesting. Why not? Why doesn't it play a role?
No, I think it
No,
does
I
play
mean,
a role.
like,
it's just not the focus of our research.
It's not the focus of our research.
Because it's functional specifically, and it can't lend itself to structural connectivity.
So it tells us very different. It gives us very different information about the brain. And so definitely there is, when studying the brain in vivo, obviously it's invaluable to be able to study the function, and particularly the functional networks that we can, the information about functional connections or correlations, let's say, in the function between different areas that we call a functional connection, that we can get only in a living subject. So it's not an either or.
Right.
So both of those things need to be put together eventually.
Right.
Yes, and I think it depends more on the application, right? Like some things are more suited for function MRI. Some other things, like TBS, are more suited, or presurgical planning for tumor removal, like tractography just helps more in those for those applications.
That's interesting. It's very interesting because we talk a lot about fMRI and compare it to diffusion MRI, and you would think that they both play a role like equally. They're both like equal in every single aspect. So we talked about surgical planning, DBS. What are some of the other clinical applications of these tools?
Everything.
Okay.
So there are actually a lot of applications where we know that a lot of brain diseases where we know that the disease is not associated with just a specific part of the brain, where we cannot point to, okay, here it is because there's a lesion there or something like that, but that are associated with networks. And so anything that, any disorder that is associated with how different areas in different parts of the brain communicate with each other, this would be useful.
Wonderful. So like epilepsy, for example, and a lot of neurological disorders. Yeah. I think one thing that is kind of really striking about the Brain Connect work is the ability to kind of develop individualized and personalized atlases. How far off are we from kind of creating individual tracks?
Yeah, that's a question we always get. If you get the perfect map of connections in this one dead brain, what does it say
when can you
about
see
the
mine?
Connections exactly in my brain? And so the answer to that is that still it's a step beyond what we have right now. Because as Chiara alluded to earlier, right now when a neurosurgeon is about to stick a sharp object into your brain, actually what they have is just a mental map of where these connections might be that she got from reading anatomy books. So we don't actually have a real map that they can navigate through. So this is the first step, getting that actual digital map that a physician can navigate through, kind of like Google Maps, where you can say, okay, how do I get from A to B? And once we better understand the relationship between that true connectional anatomy and what we see in the diffusion MRI scan, as we'll have both in these postmortem specimens, then we can try to figure out, okay, how can we get that information, how we can extract that information more accurately when we only have the MRI scan, let's say in an individual patient. So that's kind of, the next phase, I guess. I would say.
Like there would really be like no way, or it'd be really hard to step from the neuroanatomy books to the in vivo diffusion MRI without having a map in between to know for what to look in the data. So this is like the first necessary step in order to be able to do that.
It's very cool. Do you have advice for engineers coming into this space, scientists coming into this space that are excited about tractography, want to become part of groups that are working on tractography? What advice would you give them?
Tough. I don't know about advice, but I would say that I think right now things are getting more exciting. I think it's a great place to be because, in this new multimodal landscape, I think from now things will actually hopefully change. I think for a long time, tractography has, stood alone and surfed the wave of the success of being able to reconstruct these big main pathways. And it was almost too easy to reconstruct, to get those and to get them, pretty much right. But now I think to make a real, impact, we'll have to get it to the next stage. And like projects like Anastasia CONNECTS are really cool because you can now see this new multimodal environment where finally like microscopy and like diffusion MRI are finally getting together. And it's not just the technologies, but it's also the people.
Right.
And so I think transfer of knowledge and information will happen, hopefully, faster and faster, and it will get easier and easier. And so, I think it'd be an exciting time for making a change.
Definitely. What about you, Anastasia?
So advice to engineers is that you will not get far if you only see the data as ones and zeros, and that you have to actually talk to the people who understand and are able to interpret the data and the images. And that's sort of the path that I have been on. It has been a very long path from when I got my degree in electrical engineering and started here. So try to connect with the people who actually understand what's in the images rather than just treating them as, just a cube of voxels.
Okay. Well, thank you both for joining me today. I really appreciate it. It's been a wonderful conversation.
Thanks for having us.
Thank you for having us.